Operons control gene expression in bacteria. There are three elements that operons consist of. The elements are the gene that it controls, a promoter region where RNA polymerase first binds, and also an operator region between the promoter and the first gene that acts as an on or off switch. Pre-mRNA combines with SNRPs and other proteins to form a spliceosome. Within the spliceosome, snRNA base pairs with nucleotides at the ends of the introns. The RNA transcript is cut to release the intron, and the exons are spliced together. The spliceosome then comes apart, releasing mRNA, which now only contains exons. The snRNA acts as a ribosome, and RNA. The lac operon is in prokaryotes. The lac operon works when there's lactose present. First, there is the lac-I gene. The lac-I gene is a regulatory gene. The first thing that this regulatory gene does is produce the mRNA, which transcribes the mRNA that then translates into the proteins known as a repressor. This repressor is active because the lac gene is naturally off. When lactose is introduced in the bacteria, the lactose will then bind to the repressor. When it binds to the repressor, it changes the shape, and the repressor can no longer fit in the operator. The operator moves away and becomes inactive. This, by the way, is a lactose, a form of lactose. When the operator is moved out of the way because of the change in conformation, the RNA after these are made, all the lactose is broken down, and then the broken down lactose is gone, and the repressor turns active again because there is no lactose. It binds to the operator, and the lac operon is shut down. That's it. The TRP is a regulatory gene, and it creates mRNA to create the protein, which is the inactive repressor. The TRP operon is naturally on, and when it's on, it will transcribe other genes into polypeptides, which make the enzymes in tryptophan synthesis. When there's a lot of tryptophan, you don't want to keep making more because it uses more energy. That's unnecessary. The TRP will bind to the inactive site in the repressor, which makes it active. The repressor will then change its shape and move into the operator and block the operator. When it blocks the operator, the RNA polymerase cannot transcribe any other genes. Is now allowed to transcribe the genes it moves through the lac-Z genes, the lac-Y genes, and the lac-A genes. The lac-Z genes transcribe beta-galactosidase. The lac-Y gene produces permease, and the lac-A gene produces transactylase. Beta-galactylase hydrogenizes lactose into glucose and galactose. Permease is a membrane-bound cell protein that allows lactose into the cell. Lastly, there is the lac-A gene, which creates transactylase. It transfers an acetyl group from the lactase, thereby breaking it down. After these are made, all the lactose is broken down, and then the broken down lactose. Okay, if glucose levels are low, then the cyclic AMP binds to the cyclic AMP receptor protein CRP. CRP activates transcription. When glucose levels are considered enough and CAMP levels are low, then the CRP protein has an inactive shape and cannot combine upstream of the lac promoter. CRP works on several operons that encode enzymes used in catabolic pathways. If glucose is present and CRP is inactive, then the synthesis of enzymes that catabolize other compounds is slow. If glucose levels are low and CRP is active, then the genes which produce enzymes that catabolize whichever other field is present will be transcribed at high levels. That's it. Splicing itself as it regulates the passage of mRNA from the nucleus to the cytoplasm. A benefit of split genes is to enable one gene to encode for more than one polypeptide. Split genes may also facilitate the evolution of new proteins.